So this is the second video of chapter 18, cell cycle. So I said that I wanted to talk a little bit about some of the cytoskeletal considerations or cytoskeletal elements involvement in cell cycle. There's really two big ones, and then to a lesser degree a third one. The biggest ones are, one, the spindle pole is all microtubules. The centrioles are the two poles, and then the spindle is set up by microtubules, both the microtubules that associate with the kinetic core as well as the microtubules that overlap each other. And then the aster is also microtubules. And so we know that microtubules have two motoproteins that are generally associated with them, kinesin and dynein. Dynein, a minus-directed motoprotein, is going to be used at the aster to pull the spindle poles away from the center, and then kinesin, a positive-directed motoprotein, are going to be used at the non-kinetic core associated microtubules where they overlap each other to push on the spindle poles to, again, elongate the cell. And then the kinetic core associated microtubules are going to push and pull the chromosomes around until they've all aligned at the metaphase plate. And this is all going to be microtubules and kinesins and dyneins. A second lesser event is the intermediate filaments, the breakdown of the nuclear envelope and the reforming of the nuclear envelope. This is going to be lamin, which is one of the intermediate filaments. Lamin gets phosphorylated, and that causes it to break down, and that's going to be in response to one of the cyclin-dependent kinase complexes. Sets up the phosphorylation cascade that has that event synchronized with when it's supposed to happen. The last major cytoskeletal function is the actin, and of course actin is always associated with the motive protein myosin. So actin and myosin are the elements that create the contractile ring that is going to create the cleavage furrow that eventually results in the separating of the two cells, the two daughters, out of the one nascent dividing cell. The actions of the cleavage furrow and cytokinesis are performed by actin and myosin. Myosin walking down actin, causing that actin band to get tighter and tighter and tighter. Okay, so cytoskeleton is going to play a fairly important role in the phases of mitosis. So it's myosins walking down actin that causes the band to get tighter and tighter at the contractile ring, creating the cleavage furrow. It's a bipolar myosin filament, and then of course actin. And so very much like what you recognize from the sliding filament model in muscle contraction is what's going on here. But just think of it as just like the sarcomeres contraction, bringing those Z lines closer together. This is very similar, except you've got overlapping Z lines all along the contractile ring, and so that's how we get the cleavage furrow and ultimately cytokinesis. This is in animal cells, but in plant cells, because they have the cell wall, cleavage furrow wouldn't be able to divide the two cells. And so what we have instead in plant cells is Golgi-derived vesicles from when it was one cell. Those vesicles are floating around in the cytosol. And after the events of metaphase and anaphase, those vesicles will migrate toward the metaphase plate. And they will just, these vesicles will come together very much like everything that we've seen in vesicles before, where a phagosome binds to an endosome, or a synaptic vesicle binds to the cell membrane. This is going to be a very similar thing. These vesicles are budding, are binding together, popping into a larger and larger vesicle, fusing together. And since they are full of the cellulose that makes up the cell wall, they are creating a new cell wall called the cell plate. And so you can see that that plate forming at the metaphase plate. You can see that cell plate forming, and it will eventually become the new cell wall. And of course, since it's vesicles that are doing this, the cell membrane is encasing that wall, will become the new cell membrane. So it happens a little bit differently in plants than it does in animals because of the constraints of the cell wall. The way that microtubules are able to push and pull on the chromosomes is because at the centromere, the narrow region between the P and the Q arm of the chromosome, gets covered in a plaque called the kinetochore, and that plaque has a lot of proteins associated with it. Some that are preventing the activation of separase, MAD2 and BUB-R1, and others that are going to attach to the microtubules. And so we've got motor proteins in there, kinesins and dyneins, that are going to be walking down the microtubules in either direction, whether it's a growing microtubule, and so walking away from the centriole, or a shrinking microtubule, and so walking toward the centriole. And that push and pull on these chromosomes is how we end up with a alignment at the metaphase plate. And so this is going to lead very nicely to a couple of chemotherapy agents that we'll talk about in chapter 20. Anything that prevents the polymerization or depolymerization of microtubules is necessarily going to interfere with the alignment of the chromosomes at the metaphase plate. So, this is one of our ways of attacking cancer cells. If they can't get past the metaphase checkpoint, then they will have cell cycle arrest. And like I said, you either complete the cell cycle or you commit suicide. And so if we can get them to metaphase arrest, then they will commit suicide. So establishment of the mitotic spindle and kinetochore capture of microtubules is a very important part of the progression through between prometaphase and anaphase, how we get the chromosomes to align at the metaphase plate during metaphase. The events of anaphase also are very microtubule heavy. It's there are kinetochore associated microtubules, and then there are also unattached microtubules, some of which overlap with each other, and we call those polar microtubules, and they have kinesins in between them that are going to push on the centrioles away from each other, creating a lot of tension inside of those kinetochore-associated microtubules, and therefore a lot of tension on the chromosomes. So then if you cut the connections between those microtubules, you'll get a very rapid pull away from the metaphase plate by those chromosomes. And then we've also got on the unattached microtubules dyneins that are pulling on the aster, and so that's also creating that dynamic tension within the spindle. So it's really important to understand that in anaphase A, we have the shortening of the kinetochore microtubules, creating a force on the sister chromatids, and then during anaphase B, we have the movement away from each other of the centrioles due to the actions of the kinesins on the overlapping polar microtubules and the direct pull on the poles by the dyneins on the non-associated microtubules. So anaphase A and anaphase B. Anaphase A creating the tension on the chromosomes, and anaphase B pulling apart of the poles to elongate the cell. Next we're going to move on to the cyclin-dependent kinases and how we control progression through the cell cycle.